Dr. Simon Stott, our Director of Research, over to you. I can then step.
Oh, stuff is already happening for me. So I'm Simon, for those who don't know me, and I'm the Director of Research at Cure Parkinson's. I've been with the organization for 5 years, and I took on the role of Director of Research last year. And for those who are unfamiliar with Cure Parkinson's, I thought I'd just start off by discussing who we are and what we do. We are a very small research charity, registered in the UK, but we fund research internationally. We fund preclinically and also clinical research. Preclinically, it must be within 5 years of clinic, and clinically, it must be all of the research must be of a disease-modifying nature. So we don't really look at basic biology or new assays. We are focused primarily on disease modification. The charity was founded in 2005, and one of the important aspects of our work is urgency. We're very much focused on urgency for people living with Parkinson's. So how does Cure Parkinson's support the research? We have this wonderful group of people. There's a couple of faces missing here, but this is our research committee. So 4 times a year, basically every 3 months, we put a call out for research applications, and they get sent into us, and we give them to this wonderful group of people. We have clinicians, we have basic scientists, we have people living with Parkinson's who are research advocates, and they evaluate the applications for us. They send them out for peer review to get expert opinion on those applications, and 3 months later at their next meeting, the committee will decide whether to recommend or not recommend a project to be funded to the trustees. So our turnaround is about 3 to 4 months. And one example of some preclinical research that was funded last year is a project from Professor Lorraine Callier at the University of Toronto. She's looking at the potential of rifabutin, which is an antibiotic in models of Parkinson's. Previously, this research team has shown that it has interesting properties with regards to a Parkinson's protein called alpha-synuclein, and now they're investigating this more in depth. And rifabutin was also prioritized by our International Link Clinical Trials Committee just before this grant was funded, and Cure Parkinson's is now looking at setting up a clinical trial of this molecule in people with Parkinson's. So what is the ILCT committee? The International Link Clinical Trials Initiative is a large collaborative clinical trial program that is led by ourselves and the Van Andel Institute in Grand Rapids, Michigan. It involves a committee of 23 experts from around the world, and they volunteer their time. They're all very, very busy people with academic responsibilities and also clinical responsibilities, but for 2 days every year, they gather for a face-to-face meeting to discuss interesting molecules of interest and which ones should be prioritized. This photo was taken just this summer in Windsor. The origins of the ILCT committee come from one of the co-founders, Tom Isaacs, and our director of research at the time, Richard Wise, who is sitting in the third row over there. They were having lots of conversations around how Cure Parkinson's could speed up the development of new therapies for disease modification in Parkinson's. And they came upon this idea of gathering a group of experts, presenting them with these dossiers full of information about interesting molecules of interest. Interesting molecules of interest. That's good English. In 2011, they asked Professor Patrick Brundin here, who was at Lund University in Sweden at the time, if he would be prepared to chair the committee. And they started gathering this committee. They're the best of the best. They really are leading experts in the world in terms of Parkinson's. And then in 2014, the Van Andel Institute came on as a funding partner. So the meeting is held every year, either at the Van Andel Institute in Grand Rapids, Michigan, or at Cumberland Lodge here in the UK. And the committee is presented with typically 20 dossiers each dossier has everything we know about a particular molecule of interest that has shown potential for disease modification in Parkinson's. Also in the room are representatives of government regulatory bodies, the NIH, NINDS, the NIHR here in the UK. We also have stakeholders, representatives from stakeholders from around the world in terms of Parkinson's research. For example, the Michael J. Fox Foundation, Parkinson's UK, Shake It Up Australia. And importantly, we also have people with Parkinson's research advocates in the room. They play an important part in the discussion. Each molecule, there is about a half-hour discussion as to its virtues and merits. And quite often, the committee will turn around and ask members of the audience, people with Parkinson's, would you consider this as a possible optional treatment? And the opinions that are given by those people with Parkinson's quite often shift the conversation. And the committee are the only ones who are allowed to actually vote on the molecules. And at the end of the day, typically 3 to 5 molecules are prioritized for going forward into clinical trial. There's been 12 years of the program running. During that time, there have been 20 completed trials associated with the program. There's 21 ongoing trials at the moment, and we probably have 10 in startup phase at the moment, including our second phase 3 with ambroxol. This is a basic schematic of where things are at the moment with the ILCT program of trials. There's a couple that are missing on the bottom here, but this is the current state of things. And as I say, there's a lot in the planning phase at the moment. I just wanted to highlight one of the most encouraging results that's come from the ILCT program has been the GLP-1 agonists. This is a class of diabetes drug. And the work has been academically driven by Professor Tom Fultonay here at UCL. Back in 2017, the results of a phase 2B project were published. And in this study, there were 60 people that had been taking exenatide, one of these GLP-1 agonists, for 48 weeks. And 30 of them were taking exenatide, 30 were taking a placebo treatment. And what the researchers noticed, and I should point out Dylan, who's in the audience here, he was one of the principal investigators on the study. So if anyone has any questions and what I'm saying doesn't make sense, just ask Dylan. But so what they noticed when they looked at the results at the end of the study was that while the people that were on placebo, the red line here, continued to progress with their Parkinson's symptoms, the folks who were on exenatide firstly got a little bit better and then sort of stayed at that level for the remaining 48 weeks. And then during this last period, 48 weeks to 60 weeks, there is a washout period. So people are taken off drug, and we observe what happens. And this was a very exciting result. And more recently, the GLP-1 agonist field has had further encouraging results from the lixisenatide study. So this is a molecule that's very similar to exenatide. It's also a clinically used diabetes drug. And in this study, this was a much larger study conducted in France, where over 150 people were involved, and there were 23 different research sites across the country recruiting people. They were treated for 1 year with lixisenatide, and in this study they also noticed a slowing of motor progression in the people that were taking lixisenatide. That was a 1-to-1 ratio of people on lixisenatide versus people on placebo. So currently we're awaiting 2 more clinical trials that are ongoing at the moment. It's the phase 2 study in Stockholm. It's exenatide being tested there. It's in 60 people, 30 on drug and 30 on placebo for 1 year. That study has a strong brain imaging component. And then we also have the results of the phase 3 clinical trial, which should be available this time next year. Well, probably summer next year. So hopefully 2024 will provide us with a lot more information with regards to GLP-1 agonists and their potential for Parkinson's. Then we also have ambroxol, which is moving into phase 3. Ambroxol is a respiratory medication. It's been repurposed for Parkinson's. In a drug screening study, they noticed that this molecule elevated levels of an enzyme called GCase. People with Parkinson's quite often have lower levels of GCase. GCase is involved in the waste disposal of cells. And so the theory, the hypothesis supporting this work is that if we raise GCase levels, then hopefully we'll enhance the clearance of waste from cells, putting less stress on the cells and slowing their progression with regards to neurodegeneration. And this has been the case in models of Parkinson's, both in small rodent models, but also in non-human primates. They see ambroxol elevating levels of this enzyme GCase. And it led to a phase 2 clinical trial that Cure Parkinson's and the Van Andel Institute and the John Black Charitable Foundation supported. The work was, and it still is, being driven by Professor Anthony Shapiro at UCL. And in the study, we had 18 participants who took ambroxol for 6 months. And if you look at the graph on the left side there, it's the levels of GCase protein in their cerebral spinal fluid. So this is the spinal fluid that your brain sits in. And they took small samples of this and measured GCase levels in that at baseline, so before they started treatment, and then at 6 months. And what they saw is that everybody that was taking, everybody was taking ambroxol, had elevated levels of GCase. So it was basically doing what we thought it would do in terms of elevating the levels of this enzyme. And the difference between the dots here is there's a white dot and a black dot. The black dot, I believe, are people that have a genetic variation in the gene or the section of DNA that provides the instructions for making GCase. So this encouraging result has led to the setting up of the ASPRO study, ASPRO PD. It's a phase 3 2-year study involving 333 people with Parkinson's, and half of them are going to have this genetic variation and half of them are just going to have normal idiopathic Parkinson's. And the primary outcome there is going to be UPDS part 3, or 1 to 3, excuse me. So a combination of the 3. And just before I move on, before anybody asks in the Q&A, this has been slightly delayed, as Professor Bandman was saying before. Drug repurposing can be quite challenging in terms of finding the right dose and finding the right formulation of the agent being tested. In the first study, the participants were taking 21 pills a day for 6 months, every day, 21 pills. And the worst part, this was really heroic stuff, because the worst part is it's got a really bitter aftertaste. So 21 pills on top of their dopamine medication. And with the reformulation, the delay has been due to reformulation and contracting and manufacturing around that. And that's led to just a treatment regime of just 3 pills a day. So hopefully that will make life easier for participants involved in the study. Ongoing at the moment, we also have, or coming to a conclusion almost, is the ASA PD study. This has been conducted in Cambridge University by Dr. Caroline Williams-Gray. Azathioprine is an immunosuppression medication. It's used for rheumatoid arthritis primarily. And Dr. Williams-Gray and her colleagues have been treating people for 12 months with azathioprine. And there's a further 6-month period of observation. So hopefully I think the last visit is coming up very shortly. And so by early 2025, we should have the results. Starting up is an interesting study with a molecule called montelukast. This is a asthma medication that has shown to be neuroprotective in models of Parkinson's. It's also been looked at in epidemiological data, where people who have been taking large doses of montelukast have a reduced risk of going on and developing Parkinson's. So this and other supportive data has encouraged Professor Per Svenningsson at the Karolinska Institute to start up he firstly conducted a small pilot study, and he had encouraging results there. So now he is setting up a large phase 2 study that we're trying to help him to get off the ground. I'm very dry here. Please, if you don't mind. We'll continue. The also coming to an end is the Trans-Euro study. We'll hopefully have the results of this very soon. This is thank you very much, Helen. This is a cell transplantation study, a cell replacement therapy study. Ah, that's better. My voice has changed. This is a cell replacement therapy study. It was a small study that was primarily focused on establishing best practice and developing tools for much larger cell replacement therapy studies. And it was led by Professor Roger Barker at Cambridge University. And Cure Parkinson's was a supporter here. And it's now all the learnings from that study are being applied to the STEM PD project. So this is ongoing at the moment with regards to recruitment. It's a very small study. It's only 8 participants, but this is a collaboration between Professor Roger Barker and Professor Marlon Palmer, who is in Lund University in Sweden. And interestingly, this is where it gets kind of interesting. Novo Nordisk, a large pharmaceutical company that's focused primarily on diabetes, has decided that the cell transplantation field for Parkinson's has got to a stage where it's prepared to invest. So this first study is called STEM PD, and it's the phase 1 study for a new type of cell therapy. And for phase 2, there's another study called Transcendence, and that's where Novo Nordisk is going to take over and take it forward for future clinical development. So for cell transplantation, and we're seeing this a lot with cell transplantation for Parkinson's, the large pharmaceutical company in Germany, Bayer, bought a small biotech company called Blue Rock Therapeutics that are also doing a cell transplantation approach. And the Japanese stem cell transplantation project in Kyoto has now got pharmaceutical backing as well. They want to take it further in Japan and also into the US. So there is a strong trend here for pharmaceutical companies being prepared to actually back this field of research and take it forward. So if all of this sounds interesting, you might also be interested in an annual report that we put out with Dr. Kevin McFarthing, Sue Buff, Gary Rafaloff, who are all Parkinson's research advocates. And we do this in collaboration with Brian Fisk and the Michael J. Fox Foundation. We'll be coming up on our 5th year of doing it next year, and we're hoping to put out a bit of a bonanza report exploring the trends over that period of time, how things have changed. I don't need to mention EJS ACPD because George has already done a fantastic task of that, but I encourage you all to actually have a look and get involved. And hopefully during the coffee break, you also saw Emma and her poster out in the hall there with regards to the UK Parkinson's Disease Clinical Study Group, which Professor Oliver Bandman is coordinating. So finally, I'd just like to summarize by saying there's a lot of research being supported. There's very encouraging preliminary results coming through for leading to further support later stage clinical development. And as I said before, with regards to GLP-1 agonists, 2024 is a very important year. I want to finish just by thanking the Cure Parkinson's team, the research team in particular, and I'll be happy to take any questions. But to do that, what I'd like to do is have some backup. And I will ask Oliver, if you don't mind coming up and sitting on the panel. I will have Emma, if you don't mind. Georgia, if you might. Yeah, that's a surprise. And this will be a real big surprise. Dylan, I'm looking at you. Would you mind joining us on stage? Thank you. So I'm going to open it up for Q&A, and if I can't answer the question, I'm going to lean heavily on the experts here. Do we have any microphones?
Hello, is that on? Yeah. Hi, I've got a question for Dylan. You were involved in Zenotide, is that right? So could you explain how a GLP-1 agonist used to treat type 2 diabetes patients to sort of normalize their glucose levels, how that might work? What's the mechanism in Parkinson's? I speak from a former clinical researcher myself.
Yeah, sure. So there's a lot of accumulating evidence now that having type 2 diabetes increases your risk factor for getting Parkinson's. So if you have type 2 diabetes, you're about 40% more likely to get Parkinson's in your lifetime, which is overall still small risk. But we think that's due to the fact that in your brain, your neurons develop insulin resistance. They can't utilize insulin as well as the other cells in the body. And there's a lot of evidence for that, actually. So post-mortem studies of people with Parkinson's show that their neurons have elevated markers of insulin resistance. And in the lab, when you treat neurons with people, treat neurons of people with Parkinson's with these kind of drugs, you restore the insulin signaling, basically. So you restore cell survival. So in the brain, insulin isn't used to, like everywhere else, to lower blood sugar, but insulin is used to encourage cell survival and reduce cell death, basically. So anything that can restore that mechanism tends to be helpful. So that's how we think it works, essentially.
Sorry, yeah, go ahead, John.
I had a question for Georgia, actually, about the protocol. I have to say, I'm not a great one for hyperbole generally, but I think this is probably the best designed clinical trial I've ever seen. But then I have only been in research and patient care for about 40 years. I wanted to ask you whether the patient-reported outcome measures, are these simply interrogative ones in the sense of actually asking the patients to tick boxes and whatever, or are they declarative in the sense that spontaneously declarative from the patients themselves?
So I'll try my best to answer this question. I'm not a clinician by background, but there's a real range of outcome measures we're including. So the primary outcome measure that we're hoping to use at the moment is a combination of part 1 and part 2 of the MDS-UPDRS. So there's more of a focus there on the non-motor symptoms. And then we have a whole range of secondary outcome measures that we will ask participants to complete, which include questions around quality of life, mood. So there's a depression scale in there. There's a cognitive assessment, which of course is clinician administered. There is lots of others that Christina who's in the audience, who's our outcome measures specialist, might be able to remind me of. But it's a range, really. There's a whole list of them, and some of them will be kind of tick box surveys that people fill in on their own on the online database, whereas others, if they need to be clinician led or administered, they'll be done via a video call or a telephone call with a member of the delivery team. And because it is online, we want to be able to provide support for any questions or confusion around any of those assessments. So there'll be training on that database and how to fill in the assessments at the start, but there'll also be support provided if people need that help when they're completing them independently.
The lovely patient lady in the 4th row.
Thank you. I was on the phase 2 Ambroxol trial. I was one of those 18 people. I think my doctor was sort of one of the white ones in the middle. And I am very keen to be on the phase 3, and I've told other people about it. And I now get regularly people saying, I've signed up, but I haven't heard anything. Now, you've been part of the reason for that, but talking about the trying to reformat the horrible pills. But I was wondering whether there is a possibility that something like the Ambroxol, which hasn't quite started yet but is about to, might be involved in the Speedy Octopus trial or something. Is there going to be like a merge there? Because that would seem to be a positive thing?
That's a fantastic question. I think at the moment all options are on the table, and that's as much as I probably want to say.
I think there is one thing that we could add to that, though, Simon. I think it's as much as when you've got trials that have been designed and have got quite complex biochemistry needing to be done, that can make it much more complex for the EJS team, for example, if they were suddenly having to do lumbar puncture draws on people and things like that. So I think it's being very clear about when the platform is going to be really valuable and which drugs we can slot into that really easily. However, I'm with Simon. I think, let's just keep the discussion going.
It is a bit of a challenge, I have to add, to insert ambroxol if we're keen to look at a genetic cohort as well as an idiopathic cohort. The genetic cohort, to keep people blinded on that large platform would be really challenging. And if only 5% of them have this genetic variation, it'll be difficult to get them all onto ambroxol. So it would be adding complications to an already complicated project.
Have been asked by various people, have you heard anything? Do we assume that not having heard anything doesn't mean that we're not going to try?
Absolutely. And there will be communication certainly coming by the end of the year. And honestly, we would all love to be able to communicate, but going when you know that you've got confirmed facts is always a little bit more helpful, I think. I know that Suzanne's got a question online.
Yeah, this is for Georgia, please. A lot of interest in the MAMS clinical trials platform. One particular question is, how do you ensure the double blind aspect of the trials and ensuring that with different drugs being taken, you can ensure those that are on the placebo and those that are taking the active drug don't know?
So we're starting off simple, and the first treatment arms will all be matched in terms of dosage and how they're taken. So everyone will be very easily double blinded in terms of the placebo, and the two treatments that we start with will all look the same. Oliver might want to comment on this a bit further, but when we introduce new drugs in the future where it's slightly more complex and they might have different, say, dose frequencies such as UDCA, there's some very clever statistical work that can be done so that when a participant's randomized to the trial, they're either randomized to one of the arms that look the same, or they're randomized to an arm that looks different, but they still won't know if they're on the treatment or the placebo of that arm. So everyone will still be blinded. They will just be aware of kind of which treatment group they're in based on the dose frequency. But it still means that we can keep them in the trial with the shared placebo arm, because there's very clever statistical work that can be done there so that we don't have to have a huge proportion of people on placebo that's matched to that arm. It can be a smaller number. So Oliver mentioned earlier, we'd still have 400 participants taking, say, UDCA. We'd only need 100 participants on the matched placebo for UDCA, but no one would know if they're on that placebo or that treatment.
There's a question from Gaynor in the middle.
Hello. We know that there are comorbidities with a lot of people with Parkinson's. So some of the medications that you're looking at, people may already be on them. They may already have Parkinson's and be on that drug for another condition. Are we looking at that background just to have that as some information in the bank at all?
Oliver, would you like to take that?
So, short answer is it's complex, but yes, we will. So just to go through the drugs, right? So they're only of course, I will start off with UDCA. So it's the liver disease I refer to is really very rare, right? Primary biliary cholangitis. So that will not be a major hurdle in all likelihood. Only very, very few people with Parkinson's will also be on UDCA for the liver disease because they happen to have both things. I'm just using that as an example. It will be a much bigger challenge for other more widely used drugs, for example, for a drug which is widely used for the treatment of high blood pressure, temozartan, right? And And that's where we will probably encounter problems. So the current thinking is that we will exclude any patient who is already on this drug or a related drug for inclusion in the MAMS trial. And that is a problem, right? Because it does limit the number of patients we can actually use for these trials quite considerably. On the other hand, there's probably no way around it, right? So, but I need to emphasize that in parallel, people are undertaking or have already undertaken clever epidemiological studies, right? Where the number of people out there was compared to already taking, say, tolmesartan, right, for the hypertension versus other drugs for hypertension to see whether it may have a protective effect, whether fewer people who are on one of the ACT-PD feature trial drugs actually go on to develop Parkinson's disease. So
yeah, I don't know, personally, that strikes me as a bit odd that people with Parkinson's are going to be on a medication for high blood pressure anyway, because they tend to have a low blood pressure as well, don't they?
Thank you. I totally agree. And that's a considerable risk. And the same problem exists for another drug we're considering, that's terazosin. So this will be a problem, right? And believe me, so as Georgia mentioned early on, I am the co-chair of the drug selection committee, and it's always about balancing risks versus benefits. And we've considered other drugs where there would have been much more serious side effects, right? But I think for me, this is one of the key reasons why it does make sense to actually have patient-reported outcomes as a key primary outcome, because, yeah, patients, this may improve if a patient was on the blood pressure lowering medication, okay, but the patients may feel dreadful, you know, on that medication, or they may have had more falls because of blood pressure drops, etc., or other things. So, you know, this is not going to be easy. Okay, this is going to be hugely complex, and still, there will be many more hurdles to overcome. I still think this is the right way forward. I also still think, and I've said that before in sort of small group discussions, what we need to do in the next 5 years, we should not meet longer than that, hopefully less. We need to find a better way of actually selecting drugs for the next arm of ACT-PD, right? Because at the moment, and believe me, the whole working group put a huge amount of work into this. We plowed the literature, we had very lively discussions, we went over the criteria again and again. At the end of the day, it was very difficult to then be entirely certain that we've really picked the right drugs, because the jump is just enormous from, oh, let's look at the data we have from the bench, right, to, oh, what might work best in the patient. You know, this is such a gap. So what we need to have something which is between all the exciting preclinical work and the fantastic ACT-PD MAMS trial design, and that's a platform, a phase 2 de-risking platform. What I mean by that is that we compare different promising drugs, so which all work on the same mechanism in the cell. Let's just use mitochondrial function again as an example. In the best model system we have, the patient, for their biological effect. So brief 3-month trials where we compare UDCA, terazosin, nicotine, ribosamide, and for example, you know, these are all drugs which have been shown very promising results in laborious phase 2 trials. We compare them head to head, right? We use the same readouts across these different drugs. We only put patients on treatment for 3 months in the expectation that if a drug does something good, it should jolly well leave a signal in the patient's body after 3 months, okay? And then the lead compound from that de-risking platform trial, phase 2, will be taken into the next arm of the large phase 3 platform trial. Because at the moment, the gap is too big. That is, but that shouldn't stop us from doing what we're doing.
I just wondered if there is an opportunity, whether there are people with Parkinson's who are already on some of these treatments for another comorbidity, who we're not just assessing or getting that data together. Maybe it's not possible to get data.
Some of it's difficult. You're thinking more real world data, epidemiological data. Some of it's difficult.
Sorry, I'm on a real life example. I'm on azathioprine and have been for years for Crohn's disease. I don't know if anyone else is in the room, but that might be worth logging somehow. I don't know if you would.
Firstly, there is the issue around privacy and accessing medical data. But then you also have the rarity of some of the drugs. For example, refubutin is not widely used. We've explored whether there's any epidemiological data potential projects, and there's simply just not enough people using it. So there are challenges with regards to this sort of research. Can we take a question from
the
online?
Online, thank you. Yeah, this follows really what you've been discussing, but it's people have been asking whether the MAMS platform would allow for taking multiple compounds at one time. So looking at individuals taking a combination of drugs through the MAMS platform.
Oliver might be a better place to answer this than me.
No. Short answer is, this is the future. I'm convinced that in the future, patients with Parkinson's disease will be on a drug cocktail, right? So one drug will address alpha-synuclein degradation, another one will tackle inflammation, another one will tackle mitochondrial dysfunction, and so on, right? So patients will be on a combination of different drugs. But as we just all realized, it's going to be difficult enough as it stands just to get ACT-PD off the ground. Fortunately, well, hopefully, exenatide or related drug will really show its efficacy, and then very soon we may actually have exactly that scenario that patients will be put on exenatide as part of the best standard of care, right? And in addition to that, they'll be put on the new trial medication.
Actually, Adrian has got the microphone.
Hello. Well, firstly, thank you for a wonderful afternoon. I really enjoyed it. And the next question is, there seem to be lots of drugs out there already for Parkinson's people that are licensed. Is there a way that AI could help, or how do you see AI get involved with your research going forward?
Yeah. Sure. So I actually do know a bit about AI actually. So we recently published a paper using AI, but I guess the problem with AI is you need good quality data. So AI can definitely help in certain aspects when you try to apply AI to clinical data, which is I think what everyone's talking about in this room, about what drug works best. Clinical data is often quite messy and it's not really suited to some of the AI algorithms. But we have used AI recently in our IPS, our pluripotent stem cell models, where we model Parkinson's in a dish and then don't tell the AI algorithm what type of Parkinson's the cell has, whether it's got a mitochondrial dysfunction or a protein dysfunction, and the AI can look at the image and decide based on the images what type of subtype of Parkinson's that cell has. And ideally, we would expand that to use for people with people with sporadic Parkinson's, not genetic Parkinson's. So there is a role for AI, and I think there are some dedicated programs for that, but you will need dedicated combined databases and large enough databases to actually utilize AI as a tool. And I think that's a lot of what Cure Parkinson's are doing now, trying to collate all the clinical data from a lot of different cohort studies into one database, really.
Just to reaffirm what Dylan says, the quality of the data you put in is key. We had a wonderful collaboration with Parkinson's UK and Benevolent AI, and we asked their AI to go through all the medical reports on PubMed and various other sources of data, and propose to us some drugs that we could put through into the ILCT program. And we had a list of a couple of thousand, and we had no idea as to why some of the drugs were picked. So yeah, the quality of the data that's going in, firstly, is key, and then understanding why the AI is selecting things as well is really important.
And can I just answer that?
Please.
So Benevolent is funding research and etc. on motor neuron disease, right? And their clever AI algorithms identified a couple of interesting compounds which work exceptionally well in some cell culture model systems of motor neuron disease. The only problem is they're cancer drugs. Really nasty. So, you know, would we really want, as clinicians, to prescribe these to lots of patients with Parkinson's? Would you really want to take them? Probably not. And that's part of the problem when you use an approach like this. You may end up, after an awful lot of work, with some really exciting signals, but you know the drug may still not be useful or in clinical practice. Yeah.
Hi, apologies if I've misunderstood you. But if you're going to take more interest in the patient's experience, anecdotal evidence, rather than the accepted quality of life questionnaires and stuff, how do you, first of all, how do you document that? And just from experience, I'm not suggesting you do this, but some consultants will listen to what you say and then write down what they want. So that doesn't really fit either with what you're trying to do. But I do think you're trying to do the right thing. I'm just not sure how you're going to document it.
Is that with regards to the MAMS assessment? So they're all validated outcome measures that we'll be using, and they've gone through a very rigorous selection process with our outcome measures working group, and then any that we wanted further patient input on went to our PPA working group for further input. So it won't be a case. Some of them, sorry, are a bit more open than others in terms of asking how patient feels or how they feel their symptoms have changed, but they're still validated secondary outcome measures. So it's not just, sorry, I should have made that clear earlier, but they do vary in their approach. Some are, you know, very specific surveys with lots of questions. Some are wider, kind of a scale, and you kind of put your answer on, but they're all that we haven't created any for the trial. They're all ones that have already previously been used and selected by our outcome measures working group.
We'll take two more questions, one online, and we'll take Michael in the back row there. Online, please.
So, yes, of course. I have a question about nicotinamide riboside as a rescue for mitochondrial dysfunction. And any thoughts on that would be much appreciated, please.
Oliver, please.
So it's a really interesting drug. I met up with so the trial was conducted in Bergen, Norway. I met up with the team in Copenhagen at a large meeting last month. We're meeting up again here in London in November. The whole Sheffield team was then flying out to Bergen. I'm just telling you all this to see that people are actually talking to each other, and we are trying to do things together. I think we are and it is already being trialed in a larger study now in Norway. So it's definitely a compound which is worth investigating. We just don't know yet whether any of these compounds will actually slow down disease progression of Parkinson's in a meaningful way. For me, again, it would be very interesting to actually do an early proof of concept study where we combine different mitochondrial rescue drugs. Because for me, it's very likely that if we combine, say, nicotine ribosamide and UDCA, will we see more of a mitochondrial rescue effect? Will we see more of a neuroprotective effect? I hope that's helpful.
So the study that in Norway that Oliver's referring to is called the NOPAC study. It's a 2-year study involving 400 people with Parkinson's. It's a phase 2 study, and the results are expected in 2025. Michael, in the back row, please.
Yes, question about exercise. I noticed on Oliver's slides earlier, there was quite athletic looking parkies there. And more seriously, I also noticed that some of the stem cell trials are beginning to introduce exercise as a component of the next phase of the trial. Now, I would imagine it's a confounding factor for parkies mid-trial to suddenly start madly exercising. So Dylan, you must have encountered this. Oliver, you must have encountered this. How do you actually encourage patients not to change their exercise regime when they start the trial? Or do you encourage everybody to step up exercise before they start? It's very difficult. I'd love to hear your views.
So nobody's asked us about that. And remember, it was sort of at the height of COVID as well that some of the study was completed. But in my view, it would be important for people to simply not change what they're doing anyway, right? Because otherwise you have too many confounding factors, right? But you know, a worry I have is that we will only ever test any therapeutic intervention, regardless whether it's exercise or drugs, in patients who are more motivated than others, right? And I think it's fair to assume that those people, those patients who are already looking after their own body, for example, by being on a healthy diet, by doing regular exercise and so on, are then also more keen to enter drug trials, right? But, you know, it's a fact, I'm afraid, that not every patient is like that. So what we are unable to do is really to what we need to get better at is reach out to more and more patients who are, let's just call them, less motivated. I don't in any way want to stigmatize, but I think that's a key challenge. I don't know whether you agree. Or...
Yeah, I agree. I think it's important to try to keep all the variables the same, but I think at the same time, you know, some of these trials last 12 months, 18 months. It's really difficult to tell people to do exactly the same thing you were doing last year. So if anyone comes to me during a trial and says, you know, does exercise help? I would always say yes, you should do the best for your Parkinson's. And, you know, I don't think realistically anyone's going to suddenly start doing massive amounts of, you know, training for a marathon. And if the drug doesn't produce an effect over and above the effect of exercise, and I think that's also an effect that we're looking for. We're looking for a larger effect, essentially. So, you know, I would encourage people to, you know, not do anything drastic, but exercise is one of the easiest things, not easiest things to do, but one of the things that can really help long term. So I would never not advocate for that if someone comes to ask for that, essentially.